Imagine a bustling city teeming with life. Now shrink yourself down to a microscopic size and journey into the world of cells, the fundamental units of all living organisms. Like tiny cities, cells are incredibly complex and dynamic, each with its own unique structure and function. From single-celled bacteria to the trillions of cells in the human body, these microscopic marvels are the building blocks of life. In this adventure, we'll explore the intricate world of a eukaryotic cell, characterized by complex structures and specialized organelles. By understanding these organelles, we can appreciate the complexity and efficiency of cellular life. Our first stop is the cell membrane, a thin, flexible barrier that surrounds the cell and acts as a gatekeeper. This essential structure is crucial for maintaining the integrity and functionality of the cell. Without it, the cell would be vulnerable to its environment, unable to control what enters or exits, leading to chaos within the cellular environment. Imagine a bustling city wall, controlling the flow of goods and people in and out of the city. Just like a city wall, the cell membrane ensures that the cell remains a well-regulated environment. The cell membrane regulates what enters and exits the cell, ensuring that only essential nutrients and molecules are allowed in while waste products are expelled. This regulation is vital for the cell's survival as it maintains the internal balance or homeostasis necessary for the cell's functions. Without this selective permeability, harmful substances could enter the cell, and essential nutrients could be lost, disrupting the cell's delicate balance. This selectively permeable membrane is composed of a double layer of lipids, resembling a sandwich with two fatty layers facing each other. These lipid molecules are arranged in such a way that their hydrophobic, water-fearing tails face inward, shielded from water, while their hydrophilic, water-loving heads face outward, interacting with the aqueous environment. This arrangement not only provides a robust barrier, but also allows for fluidity and flexibility, essential for the cell's dynamic nature. This unique structure allows the cell membrane to be both flexible and strong, protecting the cell from its surroundings while allowing for the exchange of vital substances. The flexibility of the membrane enables the cell to change shape, move, and grow, while its strength ensures that it remains intact under various conditions. Additionally, the cell membrane contains various proteins that assist in the transport of substances, signal transduction, and cell recognition, further enhancing its role as a protective barrier and a facilitator of communication and transport within the cellular environment. Venturing deeper into the cell, we encounter the nucleus, a large spherical organelle that serves as the control center of the cell. The nucleus houses the cell's genetic material in the form of DNA, which contains the instructions for building and maintaining the entire cell. Within the nucleus, we find the nucleolus, a dense region responsible for producing ribosomes, the protein factories of the cell. The nucleus plays a crucial role in cell division, ensuring that each new cell receives a complete copy of the genetic blueprint. Surrounding the nucleus and other organelles is the cytoplasm, a gel-like substance that fills the cell. Think of it as the bustling city streets and infrastructure that support all the different parts of the city. The cytoplasm provides a medium for organelles to move around in and interact with each other. Within the cytoplasm, we find the cytoskeleton, a network of protein fibers that provide structural support and helps to maintain the cell's shape. Like the scaffolding and beams of a building, the cytoskeleton gives the cell its strength and allows for movement within the cell. Scattered throughout the cytoplasm and attached to the endoplasmic reticulum, we find ribosomes, the protein factories of the cell. Imagine bustling construction sites where proteins, the building blocks of life, are assembled according to the genetic instructions provided by the DNA. Ribosomes read the genetic code and translate it into chains of amino acids, which then fold into specific three-dimensional shapes to form functional proteins. These proteins carry out a wide variety of tasks within the cell, from catalyzing metabolic reactions to transporting molecules across the cell membrane. Extending outward from the nuclear envelope, we encounter the endoplasmic reticulum, or ER, a network of interconnected sacs and tubules that serves as a manufacturing and transport hub within the cell. This extensive network is essential for the cell's overall function and efficiency, ensuring that various biochemical processes are carried out smoothly and effectively. Picture a vast network of factories and transportation systems working together to produce and distribute goods throughout the city. This analogy helps us understand the complexity and importance of the ER within the cell. Just as a city relies on its infrastructure to function properly, the cell depends on the ER to maintain its internal environment and support various cellular activities. The rough ER is studded with ribosomes, giving it a bumpy appearance. These ribosomes are the sites of protein synthesis, where amino acids are assembled into proteins based on the genetic instructions carried by messenger RNA. The smooth ER, lacking ribosomes, is responsible for lipid synthesis, detoxification, and calcium storage. It plays a crucial role in the production of lipids, which are essential components of cell membranes and serve as energy reserves. Additionally, the smooth ER helps detoxify harmful substances and stores calcium ions, which are important for various cellular processes, including muscle contraction and signal transduction. The smooth ER's ability to detoxify substances is particularly important in liver cells, where it helps break down toxins and drugs, ensuring that they do not accumulate to harmful levels. Meanwhile, the storage of calcium ions in the smooth ER is vital for maintaining cellular homeostasis and enabling rapid responses to changes in the cell's environment. Together, the rough and smooth ER form a dynamic and versatile system that supports the cell's diverse functions. By working in concert, they ensure that the cell can produce, process, and transport a wide range of molecules, maintaining the cell's overall health and functionality. Next, we arrive at the Golgi apparatus, a stack of flattened, membrane-bound sacks resembling a post office or distribution center. Here, proteins and lipids produced in the ER are further processed, sorted, and packaged into vesicles for delivery to their final destinations within or outside the cell. The Golgi apparatus acts like a quality control center, ensuring that molecules are properly folded, modified, and labeled before being shipped to their final destinations. This intricate sorting and packaging process is essential for maintaining the organization and function of the cell. Lysosomes are the cell's specialized waste disposal system, crucial for maintaining cellular health. These membrane-bound organelles contain powerful enzymes that break down waste materials, cellular debris, and even foreign invaders like bacteria. Think of lysosomes as the cell's recycling center, where unwanted materials are processed and repurposed. They ensure that the cell remains free from clutter and potential damage by digesting and recycling worn-out organelles and macromolecules. This not only helps in waste management, but also in the efficient reuse of cellular components, contributing to the cell's overall functionality and longevity. Lysosomes play a vital role in keeping the cellular environment clean and efficient, much like a diligent janitorial staff working behind the scenes to maintain order and health. Now, let's talk about the mitochondria, often called the powerhouse of the cell. These organelles convert glucose and oxygen into energy in the form of ATP, adenosine triphosphate, through a process called cellular respiration. They even have their own DNA, which suggests they were once independent organisms. Vacuoles are versatile storage organelles found within cells, playing a critical role in the cell's ability to maintain homeostasis. These membrane-bound compartments are essential for storing nutrients, waste products, and other substances that the cell needs to segregate from the rest of the cytoplasm. Vacuoles can vary greatly in size and function, depending on the type of cell and its needs. In plant cells, the central vacuole can occupy up to 90% of the cell's volume, storing water, ions, and nutrients, and providing structural support through turgor pressure. In animal cells, vacuoles are typically smaller but still crucial for isolating harmful materials, storing waste products, and managing cellular waste. By sequestering waste products and storing essential nutrients, vacuoles help maintain the cell's internal environment, ensuring that it remains balanced and functional. They act as a buffer, protecting the cell from potential damage by isolating harmful substances and preventing them from interfering with cellular processes. In essence, vacuoles are the cell's storage units, ensuring that everything is in its right place and readily available when needed. Much like a well-organized storage room, vacuoles keep the cell efficient, functional, and prepared for whatever comes next. As we journey through the incredible world of the cell, we uncover the intricate and essential roles each component plays in maintaining life. We began with the building blocks of life: the fundamental molecules that form the foundation of all living organisms. Next, we explored the protective barriers that shield the cell from external threats while allowing vital interactions with the environment. We then ventured into the cell's control center, where the nucleus orchestrates the complex symphony of cellular activities. We looked at the support and structure provided by the cytoskeleton, giving the cell its shape and aiding in movement. Our journey took us to the protein factories, the ribosomes, where the synthesis of proteins essential for life takes place. We examined the manufacturing and transport hub, the endoplasmic reticulum, and how it works in tandem with the packaging and shipping center, the Golgi apparatus. We marveled at the protein assembly line, where proteins are folded and modified to perform their specific functions. We recognized the importance of the cellular cleanup crew, the lysosomes, in breaking down waste and recycling cellular components. Finally, we delved into the storage compartment, the vacuoles, which play a critical role in maintaining homeostasis by storing nutrients and waste. Together, these components create a harmonious and efficient system, ensuring the cell's survival and functionality. Understanding these processes not only deepens our appreciation of life's complexity, but also highlights the delicate balance that sustains it. As we conclude this journey, we are reminded of the remarkable intricacies within each cell, the tiny building blocks that come together to create the tapestry of life. Thank you for joining us on this exploration of the cell, the fundamental unit of life.